So today we're going to learn about probability and Punnett squares. So probability is the likelihood that a particular event will occur. It can be written as a fraction, a percent, or a ratio. Let's look at all those. In this picture, we have one white dot and 3 red dots. So the fraction that are white is 1 out of 4. So the fraction can be written 1/4. The percent chance that it will be white is 25%. If you're having trouble converting fractions to percents, think of how many quarters are in a dollar. There are 4 quarters in a dollar, so 25 cents would be 1 quarter, 50 cents would be 2 quarters. You get the idea. Then ratios are a little bit different. There is 1 white dot, there are 3 red dots. So the fraction and the ratio look like they would be different when they're actually the same thing, just written a little bit differently. Here's another example of writing the probability for flipping a coin. When you flip a coin, you can either get heads or tails. So if we're writing this as fractions, then 1 out of 2 chances will make it heads. That's a 50% chance, just like 50 cents is half of a dollar. And it has 1 chance to land on heads and 1 chance to land on tails, creating a 1-to-1 ratio. If you just flipped a coin 5 times and all 5 times you got heads, if you flip it again, what's the chance that you're going to get heads this time? Well, past outcomes have no effect on future outcomes. So, just because you've flipped the coin 5 times, your next flip still has 1 chance to land on heads and 1 chance to land on tails. So you have a 50% chance of having heads. Now, if we said, what's the chance of getting heads 6 times in a row? That's a different question. That's when it gets all crazy and not a very high probability. But each flip has a 50-50 chance of landing on heads. Now, you've all flipped a coin and gotten heads or tails 3 or 4 times in a row before. See, probability works best if you're predicting a large number of events. If you flip the coin 100 times, you'd have a much better chance of getting very close to a 50-50. Maybe you might get 49 times heads and 51 times tails, or 48 and 52, but you're going to be closer. If you flip the coin 1,000 times, you'll get even closer. The more times you flip that coin, the closer you're going to be to that perfect 50-50 ratio. So a large number of events works a lot better than a small number. We all know families that have 4 girls, but they had a 50-50 chance of having a girl or a boy each time they had a child. So, like we said, one family is not enough people to get a good 50-50 ratio. But if you do thousands of families, then we should have a 50-50 ratio of girls to boys. Now, in real life, it turns out that girls tend to live a little better than the boys. So yes, they are conceived at a 50-50 ratio, but there's about 51% females and 49% males over the whole world. Just a random fact. The more flips, the closer the results will be. So a dominant allele is represented by a capital letter, usually the first letter of the trait. And the recessive allele is represented by the same lowercase letter. Let me show you an example. Pea plants can be tall or short. Tall is the dominant allele. So we take the word tall and use a capital T to represent a tall plant. Then we take that same letter, T, and make it lowercase to represent short. Notice we don't switch and use the letter S. We use the same letter T for both the dominant and the recessive trait, but we make it capital for dominant, lowercase for recessive. When both alleles in the pair are the same, the organism is homozygous. It has two of the same allele. Or it's purebred. Both are the same thing. For example, two capital T's or two lowercase t's are both homozygous or purebred. When the alleles in the pair are different, the organism is heterozygous or hybrid. So this means they have two different letters. So a capital T and a lowercase t would be a heterozygous genotype. And remember, homozygous is like homosexual in that a homosexual couple dates the same sex, and a homozygous genotype has two of the same gene. Then a heterosexual couple dates a different sex from their own, and a heterozygous genotype has two different letters. Well, one's capital and one's lowercase. For example, a capital T and a lowercase t. The genetic makeup of an organism, in other words, the letters it inherited, that is its genotype. The physical appearance of an organism, the way it looks, is its phenotype. The way I remember this, genes are the genotype, physical appearance is the phenotype. So for example, the genetic makeup of an organism could be capital T, and its phenotype would be tall. Then another organism could have a genotype of capital T, lowercase t, but its phenotype would still be tall, even though its genotype was different. And then finally, it could have a genotype of lowercase t, and its phenotype would be short. We are going to learn how to do monohybrid crosses. The prefix mono means one. So when we are looking at one gene, that's a monohybrid cross. You will draw what's called a Punnett square. The word Punnett is always capitalized because it's named after Mr. Punnett, who figured this out. You'll draw a square divided into 4 quadrants, like the one on your screen. When you fill in a Punnett square, the parent alleles go at the top and at the left, and the possible offspring go in the boxes inside. The way it's always been done is that the father is put on the top of the square and the mother is put down the side. However, if you switch those up and put the mom on top and the dad on the side, it's okay. You'll still get the exact same combinations for the children. So it doesn't really matter too much if you mix them up. Let's look at how to do a monohybrid cross. First of all, figure out what the parent alleles are. So in pea plants, tall is dominant over short. So tall is a capital T and short is a lowercase t. And we want to cross a purebred tall and a purebred short pea plant. So a purebred tall parent is homozygous. Remember, homo means the same. So what are the parent alleles? Well, it's tall and it has 2 of the same, so it has 2 capital T's. So that's the first parent. What about the purebred short parent? Well, that's 2 lowercase t's. So purebred tall, 2 capitals, purebred short, 2 lowercase. Now, what are the 2 gametes this parent can make? Well, it can either make a capital T or a capital T. What about this one? A lowercase t or a lowercase t. Now, we place these parent alleles at the top and the left outside the Punnett square. They didn't tell us which was the mom and which was the dad, so you can put it wherever you need to. We have a purebred tall plant, which can either pass on a capital T or a capital T. Then we have a purebred short, which can either pass on a lowercase t or a lowercase t. Now, fill in the boxes with the possible offspring combinations. Capital letters are written first. So this capital letter can be passed on to these two offspring, and this capital letter can be passed on to these two. This lowercase letter can be passed on to these two, and this lowercase letter can be passed on to these two. So write your capital letters first. This capital letter can be passed on to this or this offspring. This capital letter can be passed on to these two offspring. This lowercase letter can be passed on to these two offspring, and this lowercase letter can be passed on to these two offspring. Finally, determine the probabilities of the different phenotypes and genotypes. All the possible genotypes are capital T, lowercase t. In fact, all 4 possibilities are the same thing, which is kind of weird. So, that's the only possible genotype, and we have a 100% of having this genotype. So all the plants will be tall. This is just like Mendel's F1 and P generation. Here are the parents, tall and short, and all the offspring are tall because they have one tall gene and one short gene, and the tall gene masks or covers the short gene. What are the possible phenotypes? How does it have to look? Oh, well, they're all going to be tall, and we have a 100% chance of that. Let's look at one more example just to see what could happen. What if we crossed a heterozygous tall and a heterozygous tall? So now we're taking these kids, the offspring, and crossing them. So this is kind of like Mendel's F1 and F2 generations. This parent can pass on a tall or a short gene. This parent can also pass on a tall or a short gene. So this capital letter can go here or here. Let's do the capitals first. This capital letter can go here or here. Now let's do the lowercase. This lowercase can go here or here. And this lowercase can go here or here. Whoa. All right, so what's going on? The possible genotypes, you can have 2 capitals, and that is tall. You can have a capital and a lowercase. That is also tall. And you can have 2 lowercase, and that is short. We have 1 of these, 2 of these, and 1 of those. So if we say how many will be tall, 3 out of 4 of them will be tall. So we have a 75% chance of having a tall plant. Then 1 out of 4 will be short, so we have a 25% chance that we'll have a short plant. Also, they might ask it a little different way. They might say, what percentage of plants have a heterozygous genotype, and what percent have a homozygous genotype? Heterozygous means the same letter. So we have 2 capitals and 2 lowercase. So 50% of them are homozygous. Then we have a capital and a lower and a capital and a lower. Those are different, so 50% are heterozygous. We'll practice more of these in the next assignment.